In this video, we will discuss about how this antigen processing of the carbohydrate structures happens and how will this lead to T cell activation. Though we have covered the T cell activation in somewhat detail in our previous videos, and also we have covered the checkpoint interaction of these T cells in that video. However, in this video, we will mainly focus on this antigen processing of carbohydrates and slightly cover this T cell activation or revise this T cell activation. So what happens in this case of antigen processing of carbohydrates is that any glycoproteins that are in the extracellular matrix through endocytosis will come within the cells, and then they will undergo endolysosomes where they will break down into different fragments, and then it will again secreted on the cell membrane as through different process. One of those process is that go they will attach to this major histocompatibility complex, and this major histocompatibility complex will express those glycoprotein on the cell surface, and these antigens will then be detected by the uh, T cells. And the other process is that this deuterionic polysaccharide process. In this case, what happens? In this case also this polysaccharide that has this deuterionic charge will go will come inside the cells through this nitrogen oxide with the help of this nitrogen oxide. Uh, However, its mechanism is different from the glycoproteins. In glycoproteins, it's come by endocytosis. In case of deuterionic polysaccharide, it comes with the help of nitrogen oxides, and it helps in moving those uh, polysaccharide within the cells, and there's major histocompatibility complex will express them on the cell surface membrane. And same thing happens, or we can say same thing with different mechanism happens for this C-type lactins. However, the mechanism for this C-type lactins, how this oligosaccharide with C-type lactins come within the cells, is not yet completely known. Uh, but uh, it has been seen that this oligosaccharide with C-type lactins come within the cells, and they will express on the cell surface, and its antigen will be expressed on the cell surface with the help of major histocompatibility complex. And apart from major histocompatibility complex 2, there is another way by which the cells can be expressed on the cell surface, that is with the help of CD1. The process in this case also remains same, except the carrier in this case is CD1 instead of major histocompatibility complex, as we can see here. And the CD1 mainly works for this glycolipid, that is lipid that has the carbohydrate structures. They will come within the cells with the help of this saposin, and then these glycolipids will then express these glycolipids will be then expressed on the cell surface with the help of this CD1 protein. And now we will briefly cover how this immune system works. So these T cells will recognize the antigens from the cell surface membrane, and then it will activate the B cells through multiple interaction. One of those interaction is through transfer this antigen from T cell receptors to the major histocompatibility complex. However, this signal is not enough to activate B cells. There need to be other signals that also needs there needs to be other signals which should be activated in order to trigger this B cell immune response. So these other signals are shown here. One of them is ICOS, CD40L, PD1L, and CD28, CD80, 86. We have covered PD1, PD1L interaction in our previous videos about the immune response, where this is considered as one of the checkpoint uh, inhibitor uh, in case of uh, in case of this immunosuppression problems, or in case of various cancers. So let us come back to the topic. So This T cells helps in activation of B cells by providing this antigen through T cell receptor to the major histocompatibility complex 2 of the B cells. And simultaneously these cytokines through this interleukin 4 interleukin 5 will also activates these B cells, helps in activation of B cells. However, in this case, if this major histocompatibility complex uh, will not interact with, will not see any antigen through the T cell receptor, then B cell activation can be done by other ways through these dendritic cells. So dendritic cells, first it will dock with the help of this HSPG. After that, the antigen will be exchanged through this April and TEC1, and simultaneously interaction of this BAFF to BAFFR will also needs to be done in order to completely activate the B cells. This is how the B cells can be activated via T cells or via dendritic cells or and via dendritic cells. So dendritic, and these T cells and dendritic cells will get activated through this extracellular and expression of this antigen via major either by major histocompatibility complex 2 or by CD1. So in this way, the antigen processing of carbohydrate structures happens, and the activations of the immune response or T cells and dendritic cells activate the how the T cells and dendritic cells activate the B cells, and that will trigger the immune response against those foreign carbohydrate structures. So that's all for this video. If you need to understand this thing in detail, you can go through the reference that has been provided in the description. Thank you for your time.